Y este acá, a lo mío, me metió por allá.
Corro yo uno.
¿Tú le pones la fecha? ¿No?
It's coming.
Hola.
Ay, lo vamos a tener. Está llegando.
Sí, es por YouTube.
¿Tiene que? I live in France.
Va a pasar.
I speak French. I speak French. I speak a little, not for a talk. It would be really.
Nosotros somos de seminario.
Hello, test. Yeah, hello. Yeah.
Somos de seminario.
Hello. I don't know. Test, test. I don't know. Welcome. ¿Quién me escogió?
Ya te digo.
It's going to be hard to.
Tengo uno amarillo.
Hold it. Okay.
And the next.
Maribel.
Yeah.
Te acabo de
Maribel.
Pero que necesito Daniel.
Yeah.
Los tres son de Mercedes.
¿Qué cuarto? ¿Ya lo entregaron?
Ella está viendo la materia.
Yeah.
Depende
Eso
de
bien.
lo que me dices.
Muchas gracias. No.
Pero cambiaron el queremos que hable. ¿Qué ves el pasaporte? No, estamos viendo este.
Yo diría que Figueroa la edad.
Laura, la rubia. ¿Dices Kardashian? ¿Estás tu espero?
¿Por qué?
Oiga, yo escuché que me estaba pidiendo para que pasara. Y la profesora le dijo como, pues básicamente lo que tienen que hacer es que se pasen todo el tiempo. También lo escuché de Violeta, pues así como por encima. No sé. Pero no fue lo mismo.
¿Cuáles son?
No se cuenta que estaba pesado y estaba virtual a por tu
¿Va a hacer listo?
¿Dónde?
¿Va a hacer listo?
¿Cuarto?
Daniel, bueno, ahí para la entrevista.
No. No, pero. Y tienes que encontrarle la S, es que tiene aquí que conexión.
¿Te copió esta?
Acuérdate que no puede ser en paréntesis. No puede. Pues lo que hace simplemente en referencias, pues el apellido, la inicial del nombre, darle tiempo al tiempo, punto, la esta.
Ok, bueno. Pero
No
todas
lo
las.
coloques en paréntesis. Lo coloques. ¿Quieres que te lea la título?
A ver,
Que todas las cosas.
pero tradúcame.
Normas.
¿Será?
Yo ni siquiera sé lo que fue la vida. pues ahí empezó a joder. Pues el primer día ya había ahí, "Ay, yo no quiero referencias".
Aparte de discusión de
Ya sí le marqué 3 veces ya.
eso,
No, no, no.
sí, creo que sí. Pues ya agrego.
Pero tocaría colocar
Sí,
como
me
la
toca.
fluorescencia rosada,
Vean.
corroborando esto con el artículo de Bruce. Sí, ok. Entonces tengamos esto acá. Conclusiones.
Ah, ok, ok. No sé. Ah, ¿cómo me representan? ¿Me asustó, profe? No le hacen examen.
¿Puedo sentarme aquí? Yo la hago.
Así que preparen las preguntas.
¿Qué?
La verdad que es preguntas. La bella.
La cara le dije yo, ¿qué? Me dice, esta hermana de unas preguntas que vamos a hacer en el seminario. Y yo le dije, y ya tienen la lista. Me dijo,
¿Se
no.
presenta el equipo? No.
Y yo, bueno, pues dígame qué fecha se va a dar aquí en el invitado. Me dice, ay, no, ¿cómo que no con usted que se dedicó
Sí.
tanto tiempo? Yo, no, pues yo le doy la manita.
Todos están.
Porque no podemos dejar morir el seminario.
Con eso. Y ahora los necesitas.
Y también como hace rato no jugaba, pues, el físico.
Pero ¿dónde está la hoja? O sea, ya pero
¿Y por qué se siente mal?
No tengo la más.
Me toca hacer la vieja,
Aló.
profesora Bruno,
o sea, nosotros le pasamos una hoja a la profe, aparte.
que no aprende nada. Yo sí soy de las que me siento primera fila.
Buenos días a todos. Queremos darles la bienvenida al segundo seminario del programa de Biología Aplicada. Y tenemos un invitado muy especial que es Timothy Tramberger. Y él es un investigador del Instituto de Investigación y Desarrollo de Francia, que es como el instituto encargado de hacer investigación en Francia. Entonces, él nos va a hablar un tema muy fascinante. Él trabaja con abscisión, las bases moleculares y genéticas de la abscisión y la maduración en frutos. Entonces, para aquellos que estaban como que les faltaban temas de biología molecular, pues acá tenemos una persona que les va seguramente a enamorar del tema. Y bueno, pues para mí es un gusto tener a Timothy acá. Hay una cosa, ¿tú vas a hablar en inglés? Ajá. Va a hablar en inglés, entonces presten mucha atención. Bueno, es todo tuyo. Gracias, Timothy.
Muchas gracias, Carolina. Buenas tardes. No, buenos días a todos. I want to apologize first for not being able to give this presentation in Spanish. I'm sorry. So I'm going to speak in English, and I'm sorry for my heavy French accent because I'm from France. So I hope everybody can understand me, and I will speak clearly, I hope, so everybody can listen to my talk about one of my favorite topics on fruit abscission. So we've been working on fruit abscission of oil palm for many years. So I'm going to give kind of an overview of the work that we've done and talk about abscission just a bit in general in the beginning, so everybody sort of understands what I'm talking about. And and then I can hopefully maybe have some questions at the end about abscission. But anyway, so first of all, oil palm is a palm species that we have a lot of genomic resources to study and to use to study. So one of the problems with why study fruit abscission in oil palm? Well, one main reason is because of there's economic losses. So there's effects on the yield and the quality. It increases cost for harvesting. The main reason is because there's a lot of labor that's needed to harvest the fruit that drop, and also the labor to identify which fruits are ready to be harvested. So the idea is to eventually have material, genetic material, that would delay would allow delay of the abscission. That means that the fruit would stay on the fruit bunch, and that would be easier to organize the harvest. Okay, so that's sort of the reason for studying fruit abscission in oil palm. And we know, I'm not going to introduce oil palm as a species. It's an economically important species. I think everybody's well aware. Controversial species, cultivated in wet areas and wet, humid areas, and there's a lot of deforestation to plant oil palm. So we're not going to talk about that, all the controversy around that. But I'm talking about a palm species that's very interesting, and we have the opportunity to study abscission, fruit abscission in a palm, because we have so many genetic resources and genomic resources. So what is organ abscission? First of all, we know organ abscission, I think most of you know it because we see leaves fall. So that's abscission. And we also know that insects or even diseases can cause leaves to fall as well, or like temporal time in the autumn, at least in the temporal regions or in the tropical regions, we have leaves that are dropping all the time. In the temperate regions, we'll have leaves dropping in the fall. That's why it's called fall, because the leaves fall. And we can also have organ abscission of flowers. So floral organ parts can drop off, like petals and tepals and sepals and everything from the floral organ, flowers, and whole flowers even. And we can also have, I don't know if I have a pointer, we can have fruit that drop. So this is just an example of an apple, which is a fleshy fruit. And we can also have dry fruit, like beans or peas, that open up and they dehisce. So they open and the seeds drop out. So organ abscission for reproductive organs, especially fruit, especially fruit, they have to be timed correctly so that the seeds are dispersed. So if the seeds aren't developed enough and the fruit drops, seeds aren't ready to germinate. So it has to be timed correctly with the development of the seed. So anyway, it's really an important aspect for distribution or dispersal of seeds. It's also a way of attracting, like the fruits are a way of attracting animals, and then that will disperse the seed eventually. So it's an important reproductive process, fruit abscission. But we have we have leaf, we have flowers, we have fruits, we have all kinds of organs from plants that drop off, they abscise. So that's organ abscission. So, and I think everybody's aware of Arabidopsis. It's a great model, molecular genetic model, which has been used a lot to explore abscission. And particularly, they've used Arabidopsis to explore floral organ abscission, that is the stamen or the sepal or the petal. And one thing that's very important about all abscission processes depend on an abscission zone. So the abscission zone, or the AZ, is the zone at the base of the organ that will allow separation between cells that will allow the organ to fall. Okay, so in any case, the leaves, the floral organs, or the fruit, or the seeds all need an abscission zone, in most cases, in almost all cases, to be activated to allow the organ to drop. So how does that work? We have an abscission zone, and in the beginning we have undifferentiated cells at the base of the organ that's developing. So at the beginning of like floral or flower development, we'll have some cells at the base of the flower that will start to develop and become what's known as the abscission zone, the AZ, and these very small cells here. So eventually those small cells will respond and acquire competence to respond to hormones or external factors of the plant to initiate abscission. And once abscission is activated, the abscission zone is competent to respond to signals, then those cells will separate, and that separation will then allow the organ to fall. So that's the basis of most all organ abscission, whether it's leaves, floral organs, or fruit or seeds. And in particular, there's a lot of enzyme, or there are a lot of hormones that are involved in either promoting abscission, like ethylene or jasmonic acid, or inhibiting abscission, like auxin or GA. And eventually, these hormones will initiate the expression of genes that are necessary to be expressed in the abscission zone to produce enzymes that will work on cell separation, that will allow cell separation to take place within the abscission zone and allow the organ to fall. So that's really the basis, the model that's been built through the years, especially at the molecular level with the Arabidopsis and also with tomato. So oil palm. In oil palm, it's a very long time from the beginning after early fruit development. This is 30 days after pollinization. Very long time to have a ripe fruit, 180 days, 6 months. And the only fruit that we see that abscises from an oil palm is ripe fruit. So it's the very late ripe fruit that will eventually fall. So that's, I've never witnessed. I know there are, I've heard of some fruit from oil palm that can abscise at an early stage, but I haven't seen this myself. But in general, in the field, what we see, only ripe fruit abscise and fall. And this happens between, it's a range, and you'll see that later in my talk. It's a range between 160 to 180 days after pollinization. So what's really amazing about the abscission zone in oil palm is you can cut the base of the fruit, and you can actually see the abscission zone with your naked eye. So you can see this. With Arabidopsis, it's impossible. It's very, very small. With oil palm, we can cut it with a knife, cut it like longitudinally, and look, and you can see it very easily. So it's amazing. And it's at the base, the abscission zone in oil palm is at the base of the mesocarp, which is the fleshy part of the fruit, and above the pedicel, which connects it to the fruit bunch. And we've done studies in Petri plates where it takes about 9 hours without any treatment, just closing it up, cutting slices of the base of the fruit, and it takes about 9 hours for cell separation to take place, which you can see here. Cells have separated on their own. So it takes about 9 hours for this to take place. So what's the problem with oil palm is that the fruit ripen in an unsynchronized way. So if you take a whole fruit bunch and you open it up, you see that these are spikelets containing ripe fruit at the top, and at the bottom they're less ripe fruit. So if this fruit bunch is harvested and then oil is extracted from the mesocarp, it's actually, you're not really optimizing the harvest because some of the fruit are not ripe and they don't have the optimal amount of oil in the mesocarp. So the idea would be that the problem is these ripe fruit start to abscise, and then the fruit bunches are harvested. So the idea is to keep these ripe fruit would actually stay on longer to allow these other lower fruit that are less ripe to ripen to increase the overall yield of the oil palm, and also help with organizing the harvest. So if we take the fruit apart, you can see that they're at different ripening stages. Just one spikelet will have different ripening stages. So these are more ripe here and less ripe here. So the first question that we asked so many years ago now was whether ethylene is involved in the process. And this is something well studied in most abscission systems, is that ethylene is a positive regulator of abscission. So we thought probably it was involved. So we first did some experiments treating different types of fruit with oil palm. So we treated young 30-day after pollination fruit, 120-day and 160-day ripe fruit with ethylene. And then we looked whether they underwent abscission. And what we found is if we treat with ethylene, only at 9 hours, the ripe fruit start to separate or size right away at 9 hours. If the younger fruit at 120 days will upsize at about 12 hours. So it takes longer. And the 30-day fruit, it takes 24 hours. So this is just showing that the abscission zone in these different stages of development are not competent to respond to ethylene in the same way. So it's the response to ethylene is developmentally regulated. You need to be ripe to be able to respond to the ethylene. And if we don't treat with ethylene, the only fruit that eventually upsize are the ripe fruit without any treatment at all. And that happens within 12 or 24 hours. So that told us that yes, ethylene is involved. Okay, no big surprise. So this is just a little video of the base of the fruit cut and reconstructed. And what it's showing is the separation. I'm showing here. This is the top here is the mesocarp. The base here is the pedicel. And in between is just the opening where it's separated already. So abscission has taken place. And these are the cells. And the yellow here at the tips of the cells, I'll play it again. I love playing this. It took a long time to make this film. So at the blue is the nuclei within the cells. The cells are elongated, and at the very tips of the cells, we see a lot of yellow. And this yellow is an antibody that recognizes polygalacturonase, pectin. Okay, so certain types of pectin. So it's telling us this, and this told us, yes, pectin is there, and we sort of knew that already, but we did this experiment to really show that yes, pectin is there, and abscission zone in the oil palm is rich in pectin. So that led us to further work because pectin is broken down. Pectin is in the middle lamella between cells. And if you want to separate cells, you have to degrade the pectin within the middle lamella. So that means that you need enzymes to do that job. And the enzyme most well known to do that are polygalacturonases. So we asked the question, are there polygalacturonases involved in oil palm abscission? We thought probably yes, because in most systems it's the case. So we looked for polygalacturonases. So this is just a longitudinal section. This is the pedicel, this is the abscission zone, and this is the mesocarp. And these are the cells here, the white, and this is lignified vascular tissue here. So if you see here in the abscission zone, so this is before treatment with ethylene, then we treated with ethylene, and we see that response, this blue part here in the abscission zone, this is a transcript. It's the expression of a gene that encodes a polygalacturonase. And this polygalacturonase is activated, the expression is activated within 6 hours. And if we look up close, we can see that it's in the abscission zone and not in the pedicel. But what we did find out was this response is quick, it's fast in the abscission zone, but it's slower. It also is expressed in the mesocarp. So it's not only expressed in the abscission zone, but it's also eventually expressed also in the mesocarp. So during ripening, this polygalacturonase, which we call EGPG4, is expressed spatially within the abscission zone and temporarily within 4 hours after ethylene treatment in the abscission zone where cell separation takes place. But it's also expressed in the mesocarp, which means that it also may play a role in the softening of the mesocarp, not specifically only in the abscission zone. So we wanted to go to another level, so we did transcriptome sequencing. So I had a PhD student, Kim, that worked on this project, and what he did was we treated the fruit. So we treated young fruit, 30-day-old fruit, and ripe fruit with ethylene, and we extracted the RNA from the abscission zone from the young fruit that don't respond to ethylene, and the ripe fruit that do respond to ethylene, and also the pedicel where there's no cell separation in response to ethylene. And we asked the question, okay, we're looking for genes that are expressed specifically in the functional AZ, that is the ripe fruit abscission zone, and we treated with ethylene at 0.369 hours, and we're really looking for genes that are specifically expressed in the abscission zone in response to ethylene. But we also, so that was the objective, was to look for abscission zone specific gene expression. So we're looking at the ethylene, the induction we're looking at ethylene-induced abscission. We're looking at the expression of genes in the abscission zone. But we also looked at the natural abscission. So this is when just fruit fall naturally. We looked at the abscission zone in those fruits. We know that treating with ethylene can cause other things. And we wanted to know, well, we want to know exactly what's happening in naturally abscising fruit. So we did both experiments looking at ethylene-treated fruit and also fruit naturally abscising in the field. And what we found was about 168 common genes that are differentially expressed. So these are genes that are increasing or decreasing in response to ethylene at 3, 6, or 9 hours after treatment. And they're also expressed in the abscission zone during ripe fruit abscission in the natural abscission, in the abscission zone. And we're happy because one gene we found was a gene we already found, was EGPG4. It came out as one of the candidates that's common between the two experiments. So that says, okay, that's very good. But we also found, in addition to the one we already found, which is sort of our control, we found 5 other genes that were very similar, had very similar sequences to genes that had functional roles in organ abscission of Arabidopsis. So that told us that our group of genes, 168 genes that we found, it's a good group. We found our gene that we already found, plus 5 genes that were already functionally characterized with Arabidopsis. So just to give an example, I don't know what happened on my picture here. Oh, that's weird. I think I have a problem with my thing, yeah. Anyway, so we found genes that were involved in anther dehiscence, floral organ abscission, seed abscission, silic dehiscence, and also with rice seed shattering. So we found genes that were already functionally characterized within these different species. So we know that at least the 168 genes that we found may have a very important role in abscission of ripe fruit. So we went further then. We wanted to basically have 168 genes. How do we validate these genes? We don't have mutants in oil palm. It's hard to validate. It's not like Arabidopsis where we have a mutant. We can look at the mutant and that validates the function of a gene. So we went back, and this work was done in Thailand. We looked at non-shedding early 120-day fruit, where the fruit do not drop naturally or shed or abscise naturally from the tree. And then we looked at 160 days where the fruit are actually shedding and abscising. And we looked at the expression. In addition, there are 2 species of oil palm. There's a species in Africa that's cultivated worldwide, and there's also a species in South America, American oil palm, and Central America, that's also used. So we looked at in the diversity, and the question was, can we find in that diversity, can we find any trees, individuals, that are like mutants that do not shed their fruit? And so we looked at all that diversity, Aelia salifera, and the hybrids between Aelia salifera, which is the American oil palm, and then the hybrid, which is Aelia guinensis, and the Lifera hybrid. And also you can have backcross. You can actually backcross the cross between the two, the hybrid, back to the Guinensis, so it's a back cross. So anyway, we had access to a wide variety of this material, looking for fruit that stay on the tree. It's very simple. And how did we do that? Well, we could observe in the field, but we also developed an in vitro test. So this test, we sliced the base of the fruit, and then we knew that with time, 24 hours, usually the fruit bases separate, and we applied a test with tweezers, opened it up, and you can see the opening here. That's a positive test. That means abscission was going to take place, or cell separation within the abscission zone took place. And then we count them. We just counted them and said, okay, we had so many, and we divided into different classes. I'll talk a little bit about that later. So basically, it was a tool that we used to phenotype. And we went into the field, and then we looked at all the different material. So at the base of on the x-axis is just different genetic material, oleifera, guinensis, American and African oil palm, the hybrids. And so as much as we could. And what it's telling us is that there's a lot of variation within the population based on our test. So like the blue is actually material that does not shed its fruit, and it doesn't separate within our in vitro test. So that was really interesting already to see a wide variation, no abscission or a lot of abscission, or a little bit of abscission. And we found one individual that didn't shed its fruit at all. So that individual we took, and we used that in gene expression studies. We said, okay, it's like a mutant. We can use our 168 as many, well, we didn't do all of them, but we did a subgroup of our 168 candidates that we found with our experiments. And we asked the question, okay, we know that they're expressed during natural abscission. We know that they're increased during ethylene treatment. The question is, are they expressed in the same way in the mutants that we found where the fruit don't shed? We predict that their expression is different. Maybe they're not expressed, or they're expressed in an opposite way. And in fact, that's what we found for a large group that we tested. The white little asterisk here, and the red is no, and the white is yes. So a lot of them, yes. So for instance, our first candidate, EGPG4, we know very well, it turned out to be also not at all expressed in the mutant, so in the abscission zone. So that validated that it probably plays an important role in abscission. So then we went to another stage and we said, okay, we know the expression in the abscission zone. We wanted to look in the mesocarp and the pedicel really to look for just specific expression in the abscission zone of a subgroup of the gene. And what we found, so we looked at the expression. This is treating with ethylene here. So this is no ethylene in the mesocarp, 9 hours of methylene, and the dark blue is high expression. And so we can see here, if we look in the abscission zone, we have expression of this group of genes here. And in the pedicel, we have very little expression. So first of all, there's very few of these genes are expressed in all the tissues, mesocarp, abscission zone, and pedicel. Only one that we tested anyway. And only these two here, are only expressed in the abscission zone. And they turn out to be polygalacturonases. So there are 2 polygalacturonases in addition to the first one that we found. There's 2 others that are specifically expressed only in the abscission zone during ethylene-induced abscission and in natural abscission in the field. So that was exciting. So we put a little model together and said, okay, so we need expression from the mesocarp. We need genes that are expressed in the mesocarp. We need some gene expression in the pedicel. And this all overlaps somewhat in the abscission zone to allow the conditions, the right conditions eventually, for cell separation to take place and for the fruit to fall. And 2 of these genes that we found are specifically expressed in the abscission zone. So we went back, go further. So we're quite happy with that. We have a group of genes. We look like many of them are validated. They function. We want to go further than that. So we asked some basic questions about abscission in general. And to do that, we modified, we improved our in vitro test. So we initiated, or we assigned what's called an abscission index. So we have a formula, and based on 4 different types of phenotypes that we can get from our in vitro test, so this is no abscission. We get very, very little bit at the beginning of separation in the abscission zone. We only have the main abscission zone that's separating, and then we have complete separation. So based on the pressure that we apply, we can have 4 types. And with those 4 types, we are with this formula, we are able to assign an abscission index to each individual that we're looking at in a population. And first thing we did was ask a question about basic abscission. So we know from Arabidopsis that there are a lot of hormones involved, and we also know that there's one very interesting pathway called the HISA-IDA pathway. IDA is a small peptide that acts as a hormone, and it binds to a receptor, HISA-like 1, and when they bind, then it initiates the expression of genes that are important for the separation, cell separation that takes place in the abscission zone. So we knew that function in Arabidopsis. This has been studied with using molecular genetics. It's very clear that it plays a role, this pathway, signaling pathway, plays a role in the induction of abscission. So we wanted to know, can we find IDA and HISA in the abscission zone? First question, so this is the thing. The IDA peptide binds the receptor and it initiates, activates genes, some genes that are involved in the abscission and cell separation. Question is, does it function in oil palm? We looked, we used sequences, we found genes in the oil palm genome, and we found that they're expressed, so this is just gene expression experiments, showing that we found IDAs from oil palm and HISA-like proteins, genes from oil palm. So, first question, yes, they're expressed. And they're induced by ethylene. So it seems like there's a link to ethylene, okay? So then we went further, we said, okay, so we know that the genes are there. We know that the receptor genes are there. We know the IDA genes are there, could function in the abscission zone. We went further, we said, okay, can we take the peptide itself? Can the peptide, if we treat the abscission zone on the slices of fruit base, can we induce abscission? And I have to say, kind of to our surprise, we looked at first the concentration of IDA. So we treated abscission zones, bases, the base of the fruit like this with IDA, and we see that we get an increase in the response with increasing concentration. So when we treat with IDA peptide, we induce cell separation in the abscission zone. And we also found that it depends on the developmental stage of the fruit. If we treat young, early development fruit abscission zone, they don't respond. These are the bars here. If we treat ripe fruit, like ethylene, like with the ethylene experiments, we get a response. So again, like ethylene, you need ripe fruit for the IDA peptide to have an effect on the abscission zone. Okay, so we're happy with that. We're like, oh, well, that's conserved. That pathway appears to be very conserved in oil palm. It's not new, but at least we say this functions in oil palm, and it's one of the first species that really we showed that. So that was exciting. So we went back again to our in vitro studies, and we used this for a different thing. This time we went, this is in Ecuador. We looked at material that's all, this is an American oil palm from Ecuador. We had access to a population, and we asked the question, again, what is the variation within that population based on the test we're using? And you can see all the little points here. This is just the abscission index, so we're using the abscission index here. And so here, too, is a lot of separation. All these points are different individuals, and they're abscising fruit. And the ones below here, -2, they're not abscising fruit. So what we found 2 individuals, and the main, and the x-axis is just different genetic backgrounds, let's say. They're different sources so it's different genetic material, each one. And this material here, we found in the same type of material, we found two individuals, one where the fruit don't upsize and another where the fruit upsize. So that's like a model. We have like a mutant and a wild type. And we decided, well, first just to show you, this is kind of amazing. And in fact, when we look at the trees, we can find that even the fruits stay on the trees and they germinate on the trees. So the fruit do not fall. They eventually germinate on the tree, which I've seen twice. I have this picture here. That's my hand. And there's another one here. So it was really amazing because I had a lot of breeders before telling me, the olifera doesn't lose its fruit. I've seen it germinate. I'm like, okay, maybe. But anyway, I did see it. It is amazing. And they remain on the tree. So this allowed us to do studies. The first question we asked was, is there a problem with the abscission zone? We know with tomato, there's a jointless mutant. The jointless mutant does not have an abscission zone, so the fruit do not fall. So the first question is maybe there's no abscission zone, and that's why the fruit remain on the tree. But in fact, when we looked at the shedding, so this is the fruit that fall normally, and the non-shedding, these are the non-upsizing fruit, the abscission zone looked similar. So it doesn't look like there's a problem in the abscission zone, at least based on these studies that we did are pretty simple. Well, we went to another level. We said, okay, let's compare at the transcriptomic level the gene expression in the abscission zone in the two species, like looking at a wild type and a mutant. So we looked at the fruit, the abscission zone of the wild type where the fruit upsize, and we looked at the abscission zone in the individual where the fruit don't upsize. And what we found was that there's a really radical difference in the expression of genes in the two different individuals, and that relate to cell proliferation. So there's an upregulation of different genes involved in cell separation, proliferation, histone or chromatin structure, and cell wall, nucleic acid metabolism, hormones, and stress. And while there's a downregulation of genes related to carbohydrate metabolism, different hormones, and different transcription factors, in addition to ROS. So we found that, there's perturbation really of gene expression, and that may be the reason that the fruit remain on the tree and we have a non-shedding individual. So that was exciting. So now that brings me really close to the end of my talk, where right now the work we're doing is combining functional genomics is really what I'm talking about when functional genomics means we're going into the genome, we're looking at the genes. I'm trying to prove that a gene has a function within a certain process like abscission. So we're using all the information that we have from functional genomic studies, transcriptome studies that we've done, and we're mixing this with genetics. So we have a population, and we're doing an analysis of a QTL analysis to look for regions of the genome that are related to the abscission process. And how do we do that? So we have a population, this is in Benin, Africa. We have a population from a single individual, that's an elite individual that produces very well fruit, very nice fruit. It's been self-pollinated. We have a population, and then again self-pollinated, and we have more population. So we have a segregating population that we can use to do our studies. And with this population, we did a QTL analysis, and we based this QTL analysis on two types of phenotyping systems. One is the in vitro test that I explained, where we're just testing to see if separation occurs or not, assigning the abscission index. And the other is actually based on the time, the day that the fruit drop, and this is based on the harvest. So every time the fruit's harvested, they mark the date, and we know that that date in terms of the pollination, because all the trees are pollinated by hand, artificially. So they know the date of pollination, and they know the date of fruit fall, because that's the date of harvest. They know, okay, fruit are falling. I forgot to mention that when people go in the field, to know that a fruit bunch is ready to harvest, they look for fruit on the ground. So basically, when there's fruit on the ground, that means abscission starts. And so that information, we saw that with that information that we have a range of variation in the days after pollination when the fruit fall from 150 to 200 days after pollination. And we also have a range of abscission index where we can see that there's differences in the way that respond in the in vitro tests. So we have these two phenotyping tests, and we resulted, after the QTL analysis, that resulted in 7 QTLs. These are 7 regions of the genome that relate to the abscission index. I think I actually mixed these up. This should be fruit drop here, and this should be abscission mix. Sorry. Anyway, I mixed these two, this one and this one up. But we have 7 QTLs that relate to fruit drop, and we have 3 QTLs relating to abscission index. So we have 10 QTLs in complete, and no QTLs correspond to each other. So it's like we're looking at two different ways of looking at abscission. One is really timing, and the other is capacity to respond based on the abscission index. It's really the capacity to respond in the abscission zone, and the other one, fruit drop, is more just timing of fruit abscission. So now, last experiment we've just done is we've looked in the population based on, we can genotype all the individuals in the population based on a microarray. So we can say, okay, this individual is homozygous for our QTL. And this individual is homozygous recessive for our QTL. So we have some genetic information about the individuals. So what we did, the question was, are there changes in gene expression of any of the gene candidates that we find within the QTL? So again, the QTLs represent a part of the genome that is responding in some way. There's some factor in that part of the genome that's allowing a delay in abscission or a delay in the way it responds in the in vitro test. So we asked the question, are there changes in any of these gene candidates in their expression in the different haplotypes where they're haplotype homozygous dominant or recessive? So 0 or 2. And we looked during a developmental time course. So we took an individual that is recessive, and we looked at developmental time course, and we looked at a homozygous dominant, and we looked at a developmental time course. We did RNA extractions of the abscission zone, and we asked the question, are there differences in gene expression? So I'm not giving the name of the genes that we found because this is, at this point, secret. So I'm just saying we found different genes that respond in significantly different ways. And they are related to, one is related to fruit drop, 2 genes related to fruit drop. So this is timing of fruit drop. And we see a large, so if we look at early, this is early fruit, 120 days, 130 days, 160 days. So these are ripe fruit. So we can see that the big bars here, this is just comparing here and here. There's big differences between here and here. This is the abscission zone of one haplotype versus another haplotype. So this haplotype, for instance, will have no delay, and this one will have a delay in fruit drop. So the main point here is there's radical, significant differences in the way some of these genes are expressed. We had about 500 gene candidates. But the fact that we had the abscission transcriptome, we knew that some of those genes we already found in our QTLs. So we looked at those genes, we really narrowed it down to about 80 genes. And now we have 4 genes. We found 2 for fruit drop, and we found 2 for abscission index, which are radically different in the different haplotypes that we tested. So these genes their expression is perturbed in these haplotypes, and the expression of these genes are important to allow abscission to take place. So if you stop or if you eliminate expression, then it'll delay the abscission taking place. And all these genes, I can say, all these genes are related, potentially related, to balances in hormone within the abscission zone. So we have nice information. These are 4 that we have. We have others as well that we're going to follow up in the future. So that's into my talk. I stopped there just to say that this has been a long story. I started in 2008, I think it was, when we started this project on fruit, oil palm fruit, and then focusing on abscission, 2 PhD projects, numerous masters and technicians and scientists in Thailand, in Ecuador, in Benin especially, people from Montpellier, from CIRAD, from IRD, from the university. So it's been it's not just me doing all this work, it's a lot of people, and we've learned a lot, and we've brought the level of really understanding about abscission, about a palm, into the realm of a model like Arabidopsis, not as high as like Arabidopsis, but it's really the monocot model for fruit abscission right now in the world. So I'm really quite happy about that. So I want to thank you for your attention. And this is just some photos of different places in Ecuador, finding the the fruit that don't fall. I was pretty excited about that. And this is the ethylene bottle, a student from Thailand, Pirapat, and another student from Thailand, Kim, and my colleague Fabian, and our group in Montpellier. This is in Thailand as well, and in Ecuador, and also in Benin. So this work has taken place in at least 3 different countries, including plus France, and with a lot of different people. And I thank you very much for your attention. Muchas gracias.
¿Preguntas?
I can even probably understand your questions in Spanish and maybe try to respond in Spanish, but it's okay. Any question, I'm happy. So.
De arriba. If the abscission zone is only flower and fruit or other parts of the plant.
Okay. So it's a good question. I'm going to answer in English, sorry. But in oil palm, we only see the abscission zone in the fruit. That's it. The leaves, no abscission. They basically eventually break. They fall. Flowers, no. They just rot. If they're not pollinated, they eventually just wither and die. Maybe they might abscise, but so far, the only real abscission zone we found in oil palm, maybe not other palms, but oil palm, is in the ripe fruit and in the fruit. Thank you.
Pero en las plantas en general,
Yeah.
Tim dijo al principio que muchas cosas se caen, como las hojas, como las brácteas de las flores.
In plants in general, you have abscission zones wherever you have an organ. So you have organs that, like flower organs, floral organs, they develop, they attract for pollination, and then once that happens, they fall, it's done. So it drops off, no longer needed. Or like fall with the leaves, their photosynthesis becomes less efficient and they need resources, so carbohydrates are taken from the leaf, remobilized, and the leaf falls. Or if there's an insect that attacks the leaf, it can respond and it can fall. Or if there's a disease, it's attacked, it can respond and fall. It helps, it protects the plant, it's important for reproduction, it serves many, many purposes, abscission in general.
Well, it is very different to work with Arabidopsis, which goes from seed to flower in 40-something days. Than like this, only the seed develop, like only the fruit develops in like 180 days.
perennial species.
perennial. So how long, like your D115 sample, so that's like, which you made the cross, knowledgeable, and how many of the descendants you have?
So these populations,
Uh-huh.
they're elite populations that are used for improving oil palm yield. And they're used for, they're actually populations to sell the seed. So these are working populations that they have they're in the field, they harvest the fruit and they sell the seeds. So we're using this population as a source of variation. That's it. And that we have the opportunity to know the date of pollination and harvest. We have that as a phenotype as well. We know that there's a
But
variation.
it's not like you don't only do experiments with it, it's also
Yeah.
it's also
We just use, yeah, it's mainly used for seed production, but we can use it for studying a character. Like I've also used it for another character, which is the acidity in the oil in the mesocarp. So you can have differences in the variation in the acidity, or we used it for the variation in when the fruit drops or how it responds to our in vitro test. So we haven't made any crosses specific to try to, not yet. That's a long term.
because how long since you planted to do experiments?
But these are individuals that are known. They already exist. So it's not like, oh, let's do this. We're going to set up this experiment. No, we couldn't do it. I'd be retired by now. So no, it's not like Arabidopsis at all. But the exciting part is now we have segments of the genome. We have markers now. If breeders are really interested in this as a character, a delay in abscission, they have the markers now already. Without us doing any more work, they have the markers. Now we want to know further. My job as a scientist is really to understand the function of genes. So I want to go further. I don't want to know just that part of the genome is responsible for delaying abscission. I want to know which gene is involved and why.
The genome of this palm is sequenced, and
Yeah.
is it it doesn't seem like it's a sample because you're on the QTL part.
Right. It's sequenced. It's been sequenced at least twice. Once was published, a Nature paper in 13, I think, 12, 13, and CIRAD, which is a French institute, also has sequenced. It's a private consortium, worldwide. It's private, so it's only CIRAD. Has not been published. So we have two genomes actually.
So how come you have not gone through QTL to what the gene is?
Well, that's that's exactly what we're doing. We do that now. I mean, the best approach would be just to sequence, like transcriptome sequence or sequence each individual to look for structural changes, SNPs, within the genes of interest. But we don't have the money for that, so we decided to do this expression study, and we found these 4 genes that are excellent candidates now for explaining why. They really explain very easily, based on their function, why there's a delay in abscission, or why there's a response in the in vitro test. So that's exciting. So we haven't published that yet. So that's why I'm not sharing the names of the genes yet.
You know it. You just
I know.
not showing us.
I'm not.
Oh my God. Questions? So just a question for enlightening our students.
Okay.
How is it for you to study with all these different people, and how can any of them, if they got interested in this type of research, can get involved?
Yeah, that's a well. I've always been a plant biologist, and because of France and the institute in France, they allowed me that opportunity to not just do plant biology, but also to interact with people from all over the world, and that's been a fantastic opportunity. And in general, anyway, science is an international effort. So, I mean, nowadays you know, it's cooperation, it's collaboration on an international level. Even if it doesn't, you know, you could be in the north or the south or whatever country, it's always collaboration. So, and that's really the social part of science is it's quite nice. I like that. So yeah, I don't know
And how they can get involved?
how they can get involved. Well, I think that's a good question. Yeah, I mean, you probably, you know, now it's the amazing thing for you, for students of this generation, is all the tools that are available. Because this work here 10 years ago wasn't possible. It wasn't possible to do anything like this with a perennial species 10 years ago because the genome wasn't sequenced, because the population wasn't. So all these things now are possible because of all the tools we have. The high throughput sequencing, the transcriptome sequencing, you name it, just all the tools that we have now, molecular tools, genomic tools that we didn't have 10, 15, 20 years ago, that now we can apply to crop species that are very difficult to study. So that's your opportunity,
Our
really.
legacy.
Yeah, that's your opportunity to go further. Because now I'm really, everything we do now is like, oh wow, okay, I don't understand. So it's like amazing. So you really have that opportunity. So thank you again for your attention.
Muchas gracias. La próxima semana los esperamos en este mismo espacio para otra conferencia.
Español.
Profe,
Porque estoy haciendo esto, si yo renuncié.
profe, Aleja, para anotarnos en la lista.
Ah, bueno. Profe, ¿puedes preparar el examen?
Ay, John Kerr. Entonces lo intento.
Ay, no.
Cuídense, nos vemos.